Okay, now we're going to have a look at what this module is about and some of the key concepts within it. So just to bring everyone up to the same level playing field, environmental monitoring is a program which evaluates the cleanliness of the manufacturing environments. It looks at the effectiveness of cleaning and disinfection programs and looks at the operation and performance of environmental controls. And the same broad principles are applicable to sterile manufacturing, non-sterile manufacturing, other biotechnological processes, and in part to the manufacture of medical devices. Essentially, any aspect of a program or process where we wish to reduce the contamination risk to the product. Arguably, environmental monitoring is one of the most complex activities that lead into the support of pharmaceutical manufacturing. This complexity does not just relate to the different techniques used, but also how one designs the program and to the difficulties that are involved in interpreting and reporting the data generated. And those of you who undertake environmental monitoring will know full well that it generates voluminous amounts of data. And it's important to draw a distinction between environmental monitoring and environmental control. Now, environmental control can be understood as the systems designed to prevent contamination, such as air handling systems and disinfection practices, whereas environmental monitoring is all about the different methods that are used to measure the effectiveness of these control systems and then the subsequent examination of the collected data. So it's always that control comes first, monitoring comes second. Okay, having outlined the basics of environmental monitoring, I'd just like to run through the core units that make up this module and course. Okay, so Unit 1 is a basic introduction to environmental monitoring. So it considers why do we do environmental monitoring? It looks at different types of contamination and where that contamination might come from. It outlines what environmental monitoring is. It describes the differences between sterile and non-sterile manufacturing and environments. Looks at regulations and applicable standards, and considers the use of environmental monitoring limits. The first unit also looks at common GMP deficiencies relating to environmental monitoring, introduces cleanroom and basic cleanroom concepts, looks at the responsibilities for carrying out environmental monitoring, and touches on the subject of documentation and record keeping. The second unit looks in more detail at the classic environmental monitoring techniques: settle plates, active air samples, swabs, contact plates and personnel sampling. It looks at the differences between the techniques, seeing which are better than others, discusses culture media and incubation regimes, looks at particle monitoring, discusses special and selective monitoring, and considers some of the latest innovations in rapid methods. Unit 3 considers the validation of environmental monitoring methods, general principles, how growth promotion regimes are constructed and designed. And then it moves on to look at the validation of active air samplers, settle plates, surface samples, risk assessment approaches to validation, in that does everything need to be validated. Then it looks at the validation of disinfectant efficacy, considers the calibration of particle counters, and looks at protocols and data review. Unit 4 discusses how an environmental monitoring program is put together and covers such important issues as the frequency of monitoring. So how often should each clean room be monitored? The locations of monitoring, So these are how are the sites for monitoring selected within the clean room. The duration of monitoring, how long should monitoring be undertaken for. How risk-based approaches can help us with the program design. It then considers how microorganisms are dispersed within clean rooms and how they adhere to surfaces and how that affects the recoveries that we get from our environmental monitoring techniques. It then looks at clean room operational state. So for instance, should the monitoring be done at rest or the in-use condition? It also looks at particle monitoring or the monitoring of non-viable contaminants and how cleaning and disinfection can inform these aspects of environmental monitoring design. Unit 5 looks at special types of environmental monitoring, and in particular it looks at things that aren't covered by the regulatory guidances. So how do we deal with those aspects of the programme that aren't so clear-cut? It also looks at the time of sampling. So for instance, does a worst case of environmental monitoring exist? It also looks at incubation times and perhaps how we set those parameters and the maximum incubation times we wish to incubate samples for. It looks at sampling methods and considers parametric release. Now, parametric release is something which is applied to terminally sterilised products in lieu of sterility testing. And here, environmental monitoring data informs us as to whether or not we can release a terminally sterilised product. The unit also then looks at a case study, the environmental monitoring of a sterility testing isolator, gives some guidance on how different disinfectants can be selected, discusses and debates when anaerobic monitoring is necessary and under what circumstances it might be required. It also looks at the oft neglected area of the microbiology monitoring of microbiological laboratories, looks at aseptic filling, discusses media simulation trials and staff training. Unit 6 considers special types of environmental monitoring and considers such phenomena as how are microorganisms distributed within clean rooms and how does that inform us about how we should go about taking samples. The use and setting of alert and action levels, how often these should be calculated, how should trend analysis be undertaken, and how are control charts undertaken and statistical techniques formed. Unit 7 considers the microorganisms that are actually found in clean rooms. So this looks at the common microflora and their origins, how microorganisms survive in clean room environments, what factors affect the recovery of microorganisms. It looks at microbial culture media and the types of microorganisms that should be used to challenge, identification techniques, trending, and also looks at investigations and risk assessments. And this is looked at in particular in light of aseptic processing. Unit 8 takes the risk assessment approach further and offers a whole variety of tools and techniques that are mathematically and scientifically grounded. It also looks at the control of microbial contamination, looks at the practical application of risk assessments, particularly the hazard analysis and critical control points technique and the failure modes and effectiveness assessment methods, the FMEA, and various tables. It also looks at methods for carrying out of specification and out of limits reporting. Unit 9 considers the classification of clean rooms and clean zones, how these are done formally, particle count classification, the measurement of other physical parameters, the special requirements for unidirectional airflow devices, what used to be called laminar airflow cabinets, and more on risk assessment and troubleshooting in relation to particle counting. Unit 10 is the last unit of the course, and this looks at the various exercises and presents the answers and is really a revision and learning techniques module. Okay, the next part of this presentation, I'm going to have a look at some of the key concepts that will help you throughout the course, and if you've already read the module, may serve as useful revision aids. Okay, the first thing is about standards. So there aren't very many standards for environmental monitoring for the microbiologist to draw upon. Those that are there are within Europe. There's EU GMP Annex 1, which gives some information for the manufacture of sterile products. So if you're involved in aseptics or terminally sterilised products, then there's a little bit of guidance here. Then there's the USP, Chapter 1116, which in 2011 changed to a chapter that's now only involved with the manufacture of sterile products. Then there's the FDA Guide to Aseptic Processing, which was last revised in 2004, and again only really deals with sterile products. Then there are various Code of Federal Regulations, such as CFR 211.42. Then with the ISO, the International Standards Organisation, there's ISO 14644, which helps with cleanroom classification, only really deals here with particle counting, and ISO 14698, which deals with biocontamination control. In my view, not the most valuable of standards, but there's a few things in there of interest. So really, if you're involved in non-steriles, you haven't really got a lot to turn to. If you're involved in steriles, there is some guidance, but there's still a lot of work to do. Okay, in terms of the common methods used for environmental monitoring, these are on the screen now what we describe as the classic methods. We have to assess the microbial content of the air. We have active or volumetric air samplers, which take in a cubic metre of air. And these either work by centrifugal forces, by impaction or filtration. Then there are saddle plates, and these are designed to show what particles fall out of the air due to gravity, and these particles may contain microorganisms. So they're really of value if they're put in the right place. Then there are particle counters. In terms of surface monitoring, there's the contact plate, or sometimes called RODAC in parts of North America. RODAC is an acronym for Replicate Organism Detection and Counting. And then there are swabs. And then for people, there are the classic finger dabs and the plates of people's gowns or cleanroom suits. Particle counters, so I'll say a little bit more about that technology. But a particle counter is a device which sizes a particle, and basically it works, it has a special photodiode inside it, and it detects the amount of light scattering when a particle passes through a laser. And that degree of scattering determines both the size and the number of particles that pass through the laser. And these counters are relatively accurate. They're calibrated using latex spheres of a certain size and number. One of the big innovations in the last few years is with rapid method for environmental monitoring. And here the spectrophotometric particle counters are the main device to appear. And these are a bit like classic particle counters, but they not only count the particles, they can differentiate as to whether those particles are viable, as in related to a microorganism, or are inert material. Now it's all based on microorganisms possessing organic autofluorescent compounds which the microorganism needs for its own cellular function. So for example, NADH, riboflavin, or with spores, DPA. Now when these so-called elements of the cell are excited by a laser, they autofluoresce, and these technologies can detect that and register that there is a viable event. Now the limitation is that these viable events, which are expressed as bioluminescent light units, don't actually relate to a CFU. So they tell us there's something there, but they're less good at telling us how much and what it was. Also, it's important to stress at the moment this technology is somewhat in its infancy, and some of the counters are prone to fluoresce to certain fibres that might be shed from cleanroom suits, or even to alcohol sprays. I think they will become the future one day, but at the moment they're useful, but they cannot replace the classic methods. Each of the environmental monitoring methods that I've outlined requires validation to show its suitability within the cleanroom environment. And here we have a chapter in the United States Pharmacopoeia, chapter 1223, which is useful. And it does give an element of guidance in how to go about validating some of these methods. And it's particularly useful for when we wish to implement rapid and alternative methods. And you can see from the quote there that the USP is very much driving the user in this direction. Okay, an important area of method validation is drawing up a protocol. So you always need to have a protocol before you go about undertaking the validation, particularly for putting forward the acceptance criteria, and this is something which particularly FDA would be very keen on. So I've put up on the screen the important elements of a method validation protocol, and you can see on there some of the important things that you'd wish to include, such as what guidelines you're following, what types of samples you're going to be testing, how many samples, where are those samples going to be taken from, whether you're going to use any reference preparations. Now this might include the use of challenge microorganisms, and for instance where you're going to get those microorganisms from, what particular type of culture collection. Reagents and media, incubation parameters, very important for environmental monitoring, and how are you going to analyse the data and interpret it and work out whether your study has been successful or not. Another area to touch on is limits for environmental monitoring. Now for non-steriles there are no limits, it's entirely down to the user to draw those up on the basis of risk and drawing on a statistical technique. With sterile areas, EU GMP and FDA do set action levels, but they expect the microbiologists to set their own alert levels, and that's based on historical data. Now, to turn things on its head, the new edition of the USP 1116 chapter has no limits for environmental monitoring. Instead, it looks at contamination rate metrics. So it's more concerned about how often does a non-zero event occur, or how often does a count occur. It doesn't matter what the count is, just how often it happened. And that's a completely different way of assessing data to the classic action level techniques. So I think it's important to emphasise those, and it's important to read that section of the module that explains those more fully. Okay, so there's an exercise on the screen, and I'd like you to take 5 or 10 minutes to write down in groups, if you prefer, what are some of the weaknesses associated with environmental monitoring. So if the person who's in the room playing this video, if you could pause it, go away, do the exercise, and then perhaps one or two people could act as spokespeople for the class and report back, and then we'll resume the video and I'll give you my thoughts, and we can see whether some of the things I've thought of are the same as some of the things that you've thought of. Okay, so I'm pleased to be back with you, and I hope you enjoyed the exercise. I'll move on to have a look at some of the things that I thought were the weaknesses in environmental monitoring. So one of the weaknesses is that the results from the monitoring are not available for several days after the event due to the long incubation times. So we're always looking back, we're always looking with environmental monitoring at something that happened, say 5, 7, even 10 days ago. Another weakness is that we can only look at certain periods of manufacturing because we've only got so many techniques and we've only got so much time and we can't capture everything. Another weakness is that the results are often not reproducible. So if we get a high count and we go back a week later, the chances of getting a high count in the area again are relatively slim. It's often the individual results don't tell us very much, and we need to put a lot of those results together in terms of a trend. And it's really the long-term trend that informs us what's going on in the environment. There's also weaknesses with the types of agar. If we use different agars, we get different results because different microorganisms like growing on different agars. There's also different incubation conditions that will encourage different types of microorganisms to grow at different rates. So here we have temperature and time. Then if we find something growing on a plate, the identification method we use and the database that forms that identification technique may well give us a different result. Another factor is, in terms of recoveries, how well we've neutralised any disinfectant residues that might be, say, present on the surface. There are further weaknesses that you will probably have had in your notes as well, so it'd be useful perhaps to compare those two sets. Okay, now let's change subject and move on to monitoring plans. And these are some important questions. So if you're writing an environmental monitoring plan, here are some of the things that you ought to be considering in that plan. So for example, how frequent should monitoring be? How long should monitoring last for? How are you going to select the sample site locations? What kind of maps will you include? Should your monitoring plan reflect cleaning and disinfection practices? Do you go monitoring straight after cleaning or just before? Or would you go at any time of the day? You need to describe the sampling procedure and sample handling. You also need to describe the sample incubation regime, outline the methods for data analysis, outline the investigative responses when you exceed action levels, include detail about the establishment of alert and action levels, and describe who will do the sampling. The module also looks at special types of environmental monitoring. So there'll be a section on media